Hello students, this is Chandrasekhar, Zoology faculty from Sri Chaitanya. In Sri Chaitanya's NEET Sankalp 2.0 program, today we are moving forward with a new chapter called Evolution. As per the NEET weightage, 3 to 4 questions on an average they are asking from this chapter. So let us get into the topic. What is evolution? Evolution is the branch of science that deals with the history of life. That means it deals with the origin of life, it deals with the diversity of life, and also deals with the continuous genetic adaptations of living organisms through geological time scale. And what are the topics that are going to be covered under this evolution chapter? Origin of life, evolution of life forms a theory, evidences in favor of evolution, adaptive radiation, biological evolution, mechanism of evolution, Hardy-Weinberg principle, and brief account of evolution, and finally origin and evolution of man. So the first topic is origin of life. Before explaining the origin of life, we have to talk about origin of universe here first. And what is the measurement that can be used to measure the distance between stars? Light years, stellar distances are measured in light years. That means whatever the distance a light travelled in 1 year can be calculated. Based on that, they will mention the distance between the stars. Next topic is origin of universe. Origin of universe is explained by a theory called Big Bang theory. This origin of universe happened nearly 13.8 billion years ago. Regarding the origin of universe, see this process. There was a mass of energy initially. In that mass of energy, huge thermonuclear explosion happened. Because of thermonuclear explosion, it was expanded. The temperature in that slightly decreased compared to the previous stage. This expanded portion is called universe. It has happened 13.8 billion years ago. Once the universe is formed, hydrogen and helium were formed after the formation of universe. This hydrogen, helium undergone condensation due to gravitation led to the formation of galaxies. Several galaxies formed due to condensation between hydrogen and helium. Among those galaxies, one of the galaxies is Milky Way galaxy. In that Milky Way galaxy, soon the solar system developed. In the solar system, the planet Earth were formed. Earth was formed nearly 4.5 billion years ago, but universe formed nearly 13.8 billion years ago. In this origin of a life topic, see this. The origin of life is considered a unique event in the history of universe. The universe is very vast. Relatively speaking, the earth, if you see in the universe, it is a small speck, a very small content. Next, regarding light years also, I want to tell you a point. See this point. What we see today is an object whose emitted light started its journey millions of years back and from trillions of kilometers away and reaching our eyes now. So whatever the light you see that is coming from the star today, it was not emitted today. It was emitted some millions of years ago, travelled trillions of kilometers. So that is the reason why when you see the light coming from the stars, it is like we are looking into the past, not we are in present. But if you see the light that is coming from the objects that are present in our immediate surroundings, that is present only. But the light coming from the stars is nothing but looking into the past. So in the origin of life, we have several theories. So regarding the theories, let us discuss the theories one by one. Important point, let me underline here. Big Bang theory. Huge clusters of galaxies comprise, what is there in the galaxies? Stars and clouds of gases, dust that is present in the galaxies. And the kind of explosion that has happened is unimaginable. Very huge explosion happened. Hence it is said to be Big Bang. And Big Bang explains only about the origin of universe, not related to the origin of life. Now let us discuss about the theories related to origin of life. There are many theories explaining the origin of life. The first theory is called special connotation theory, proposed by Father Soraj, completely a mythological belief. According to special creation theory, there are three connotations are there. The first connotation in the special creation theory is the life that we see today were formed as such. The second connotation is diversity was same since past and going to be same in the future. And the third connotation according to the special creation theory is the age of earth is around 4,000 years. So 3 points of special creation theory: all living organisms that we see today were created as such, first point. Diversity was same since creation and will be same in future, point 2. And age of earth is 4,000 years. This is according to a mythological belief. This is one theory related to origin of life. Second theory related to the origin of life is panspermia, also known as a spore theory, also called cosmozoic theory. So what is a panspermia? According to this theory, the life was already there in the universe in the form of resistant spores. Those resistant spores are called panspermia. They migrated onto the earth along with the cosmic dust. This is the reason for the origin of life on the earth. So still it is a favorite idea for astronomers. It was described by early Greeks, but it is still a favorite idea for astronomers. So this theory is called panspermia. Next theory is theory of spontaneous generation. Theory of spontaneous generation. According to the theory of spontaneous generation, which is also called a theory of abiogenesis. See the points. So this theory was followed by the philosophers like Aristotle, Thales, Plato, von Helmont. According to this theory, see this, for a long time it was also believed that life came out of decaying and rotting matter like straw, mud. That means life came out of dead. So this theory is called spontaneous generation theory. Next theory is theory of biogenesis. According to biogenesis theory, life comes from pre-existing life. Abio is saying life is coming from decaying and rotting matter. Biogenesis theory is saying that life comes from pre-existing living organisms. So this theory of spontaneous generation, abiogenesis theory, was disproved by Louis Pasteur and other scientists like Francesco Redi and Spallanzani by different experiments. They disproved the theory of spontaneous generation. Here I would like to discuss about the experiment that is conducted by the Louis Pasteur, that is called Swan neck flask experiment. In Swan neck flask experiment, he took sterilized flasks free from microbial contamination, and into the flask he took boiled meat broth, or you can also say dead yeast. Then he closed one flask and he kept other flask open. After few days, when he observed, in the open flask, microbes which are present outside entered into the flask. By using that boiled meat broth as nutrients, they multiplied and produced new microbes. That means this flask which was opened is explaining life came from pre-existing life. The pre-existing microbes entered, they multiplied by using the dead yeast as nutrients. But what happened in the closed flask? He did not see the growth of any microbes in the closed flask from the boiled meat broth. So that saying nothing come from dead. So the closed flask is saying that nothing come from dead. Open flask is saying that life come from existing living organisms. This is the Swan neck flask experiment explained by the Louis Pasteur. So special creation theory, panspermia theory, spontaneous generation theory, biogenesis theory. So finally the important theory is theory of Haldane and Oparin, which is also called theory of biochemical evolution, also called coercive theory. This is the theory that is widely accepted. So what is this Haldane and Oparin theory is stating? According to Haldane and Oparin theory, life come from pre-existing non-living organic molecules. Life could have come from pre-existing non-living only but organic molecules. So, initially inorganic molecules become organic, organic to life with the action of physical forces. So, this was explained by Haldane and Oparin. Haldane is of England, Oparin is of Russia. They explained this theory, and they have explained this theory in two versions. First half of the theory is called chemical evolution, that is chemogeny. Second half of the theory is biological evolution, that is biogeny. First let me explain about chemogeny. When the earth was formed, it was formed around 4.5 billion years ago. So, at the time the temperature on the earth was around 5000 to 6000 degrees centigrade. The earth was in the form of molten mass. That means a very hot ball with molten rocks and metals. This molten mass cooled down, and it took millions of years for cooling down, but this molten mass released some amount of components surrounding the environment of the earth. There was no environment at the time, but some components are released by the molten mass into the atmosphere, like ammonia, methane, carbon dioxide and water vapor. So, they surrounded the earth and they formed a primitive atmosphere. This primitive atmosphere was reducing atmosphere. There was no free oxygen at that time, and temperature was very high, volcanic storms were there, reducing atmosphere were there initially when the earth was formed. So, what happened into these components that are surrounded the earth? UV rays acted upon the water vapor. They split the H2O into H2 and O2. H2 being lighter escaped, but O2, which is produced in this particular reaction, it immediately reacted with ammonia and methane. The reaction between oxygen with the methane produced more amount of carbon dioxide. Oxygen with the ammonia produced more amount of water vapor. In this reaction, of course, ozone is also produced, O3. But what happened when more amount of water vapor and carbon dioxide is produced? Carbon dioxide and water vapor together formed clouds. Those clouds condensed. Condensation of clouds resulted in rain. That rain of several years slowly cooled the earth and also established water bodies on the earth. So, this is the water body on the earth. So, many components from that atmosphere ran down into this water, like ammonia, some hydrocarbons, methane, cyanides, etc. This water is said to be the prebiotic soup because life is going to come in this water. Haldane called it as hot dilute soup. So, what happened in these components in this water? Still the water is very hot only. So, the heat which is present in the water provided energy for several reactions in this water. Like ammonia, hydrocarbons, methane, cyanides, they undergo oxidation, reduction, condensation reactions. In those reactions, finally some monomers are formed. Amino acid is a monomer, simple sugar is a monomer, fatty acid is a monomer, nitrogenous bases are monomers. They slowly formed nucleosides and finally nucleotides. So, all these are monomers. Monomers are nothing but chemicals, they are organic monomers. But there is a polymerization reaction in these monomers. That means several amino acids joined to each other by peptide bonds can able to form proteins. Protein is a polymer. From monomers, polymers formed in this water. Next, simple sugars finally formed carbohydrates like polysaccharides. Fatty acids to fats. Nucleotides slowly formed nucleic acids along with some proteins formed finally nucleoproteins. So, this is chemogeny, formation of chemicals. So, this is the first half of the theory of evolution by Haldane and Oparin synthesis. So, chemogeny is followed by biogeny, but let me tell you before going for the biogeny, this chemogeny was experimentally proved by a scientist called Stanley Miller in 1953. So, Miller conducted this experiment in 1953, Miller's experiment. So, what Miller did to prove the chemogeny? See this. Miller took a spark chamber. Spark chamber with some gaseous composition is to imitate the primitive atmosphere explained by the Haldane and Oparin. So the gaseous composition of a spark chamber includes methane, ammonia, water vapor, hydrogen. Hydrogen particularly they took to say it is a reducing atmosphere. Next, for the physical forces like UV rays, he took some tungsten electrodes, created spark, and the temperature in the spark chamber is around 800 degrees centigrade. Then the spark chamber is connected to condenser, condenser connected to liquid water trap, liquid water trap connected to the boiling chamber again connected back to the spark chamber. So what happened in this experiment? So by creating the spark with the help of electrodes, he created physical force. In that physical force, there were some reactions between these components present in the spark chamber like methane, ammonia, water vapor and hydrogen. So what happened? After the reaction, they allowed these components to pass through the condenser. While the components are passing through the condenser, they were cooled down. So condenser is nothing but a kind of imitation of rain of that day, and electrodes is the imitation of physical forces. Now the components are passing through the liquid trap. If any compounds like monomers are formed between the reaction in these components, they will be collected into the liquid trap. If there are no monomers are formed, again those components will go back to the boiling chamber. By heating it, sending it back to the spark chamber, one more cycle of reaction. So he is conducted this reaction for many times. One fine day when he opened this liquid water trap, the Miller found few amino acids like glycine, alanine, aspartic acid. So other scientists by similar experiments, they observed the formation of simple sugars, nitrogenous bases, even some pigments also. So Miller did not find all those. Miller found amino acids like glycine, alanine and aspartic acid. So this is Stanley Miller's experiment. In this experiment, we came to know that monomers are formed, and monomers when they are formed by polymerization, they can form the polymers. That means Miller's experiment is explaining the chemical evolution, chemogeny. Chemogeny is followed by the biogeny. See, organic monomers to polymers is chemogeny, and chemoheterotroph formation is the first life. But between these two, there is a component called protobionts. So what are protobionts? Protobionts are also called coacervates. How these protobionts are formed in this water? Protobionts are formed in this water by the intermolecular attractions between these organic molecules. Protobionts is not life, but that is pre-life with some limited metabolic activity. What are the characteristics of protobionts? Protobionts are partially isolated from the surroundings. They are formed by intermolecular attractions. They maintain internal environment, but it is not life, so it is unable to reproduce. Next, it is not considered as life because of one more point, due to the lack of a definite plasma membrane, but they show some limited metabolic activity. So these components are called protobionts. From protobionts, the first form of life started. The first form of life that is produced from the protobionts is called chemoheterotrophs, also called anaerobic heterotrophs. That is the first life on earth. From chemoheterotrophs, slowly chemoautotrophs evolved. What is the difference between chemohetero and chemoauto? Chemoheterotrophs, so they simply absorbed glucose from the surrounding water and produced energy by fermentation in anaerobic process. But chemoautotrophs, they can able to synthesize their own carbohydrates by using the energy due to the breakdown of chemicals. Slowly chemoautotrophs evolved into anoxygenic photoautotrophs. They can fix the sunlight and they get energy, but they did not produce any oxygen in that reaction, hence they are said to be anoxygenic photoautotrophs. Purple and green sulfur bacteria are the examples. And finally anoxygenic photoautotrophs evolved into oxygenic photoautotrophs like blue-green algae. Oxygenic photoautotrophs can able to release oxygen by splitting the water molecule, and also energy required for this reaction is provided by fixing the sunlight. Once the oxygenic photoautotrophs evolved, now the oxygen started released into the atmosphere. That led to the evolution of aerobes. Till oxygenic photoautotrophs, they are all prokaryotes. From oxygenic photoautotrophs, unicellular eukaryotes evolved. Slowly from unicellular eukaryotes, multicellular eukaryotes evolved. So this is about biogeny. See, biogeny precedes chemogeny. Chemogeny is followed by the biogeny. So this is the theory explained by Halden and Oparin. And what was the composition of methane and ammonia and hydrogen taken in this spark chamber? It is around 2:1:2. Methane 2, ammonia 1 parts and hydrogen is 2. So, this is about the theory of biochemical evolution. So, universe formed around 13.8 billion years ago, earth formed around 4.5 billion years ago. But when do the life started on earth? Life originated 0.5 billion years after the formation of earth, that is around 4 billion years ago. But 3 billion years ago, first non-cellular forms of like viruses, they have started on earth. They are in the giant molecules of RNA, proteins and polysaccharides. That is first non-cellular form of life in the form of virus with giant molecules of proteins, polysaccharides and carbohydrates. Later, for about 2 billion years ago, cellular forms of life started on earth. See this version of a biogenesis, the first form of life arose slowly through evolutionary forces from non-living molecules, is accepted by majority, this theory. However, once formed, how the first cellular forms of life could have evolved into the complex biodiversity of today is the fascinating story that we will discuss below. Next topic is evolution of life forms theory. Here I have already discussed about special creation theory, connotations. The life that we see today were created as such. Diversity was same in the past and going to be same in the future. Age of earth is 4,000 years. This special creation theory was opposed by Darwin and Alfred Wallace. Darwin he wrote the natural selection theory, but Alfred Wallace is said to be the co-author of natural selection theory, because Darwin and Wallace, they come to the conclusions during the same time but not from the same place. Darwin's most of the observations from Galapagos Islands, Wallace most of the observations from Malay Archipelago, Asia. But same conclusions during the same time from different places, hence the Wallace is said to be the co-author of natural selection theory. And in what way the Darwin opposed special creation theory? See this point. Darwinian theory I will explain later. HMS Beagle and theory I will explain later, but only this point of Darwin I would like to discuss. Charles Darwin concluded that existing living forms, that means the organisms which are there now, share similarities to varying degrees, not only among themselves, also with the organisms existed millions of years ago. That means present organisms are evolved ones. Sharing similarities with the organisms existed millions of years ago is nothing but extinct organisms. So what Darwin said, there is evolution, there is extinction, and since millions of years it has happened. So that finally saying age of earth is not 4,000 years, it is some 4.5 billion years, because life extinctions, evolutions are happening since some millions of years. And other points of Darwinian theory I will discuss separately in the theories topic. And now let us talk about evidences in favor of evolution. The first evidences are evidences from paleontology. Paleontology is the study of fossils. Next, what are fossils? Fossils are the remains of hard parts of life forms found in rocks. How do the fossils are formed? Rocks form sediments, and the cross section of these sediments shows the organisms or body parts of past organisms. So the sedimentary rocks are the main sites for the formation of fossils. The hard parts of dead organisms are called fossils. Okay, fine. What are these fossils explain? Fossils are said to be the documentary evidences. That means they can able to show an organism existed millions of years ago prior to the human evolution. Next, fossils based on their presence in different layers of the earth, they can explain the sequence of evolution of a particular organism. Fossils can explain connecting links, the organism showing the characteristics of two different groups that are existed in different periods. Sir, what is a period here? So for our convenience, we have divided the age of earth into some eras, eras into some periods, periods into some epochs. What are the eras of earth? So earth is divided into three main eras. One is Paleozoic era, second one is Mesozoic era, the third one is Cenozoic era. Paleozoic, Mesozoic and Cenozoic eras. See this? These are the eras. And in the Paleozoic era, there are some periods like Cambrian, Ordovician, Silurian, Devonian, Carboniferous, Permian. In the Mesozoic era, the periods includes Triassic, Jurassic and Cretaceous. And in the Cenozoic era, the periods includes Tertiary and Quaternary. Tertiary and Quaternary. So, Earth of the age of Earth, that means Earth is divided into eras, periods, epochs, depending on the different rocky layers. And how can you calculate the age of a fossil? The most common method used to calculate the age of fossils is radiocarbon dating. In this, by using the half-life of C14, we can able to calculate the age of the fossil. Along with the radiocarbon dating, the age of fossil can be studied by using uranium lead method, by using potassium argon method, by using electron spin resonance method, which is more accurate. So, generally the radiocarbon dating method is used for the fossils that are less than 45,000 years old, and potassium, uranium lead, potassium argon methods, electron spin resonance methods can be used to know the age of fossils that are more than 45,000 years, and electron spin resonance is the most accurate method. Most accurate method. Okay. See some more points related to the fossils. I already told you, like documentary, sequences and explains connecting links. Ocean is the active site for fossilization compared to the land, because the salt which is present in the ocean can prevent the decomposition of the dead organism. Next, dinosaurs are generally, we were not there while dinosaurs existing on the earth, but still we can able to picturize the dinosaur depending on it, on its fossil availability. On its fossil availability. In the next page there is a picture, let me tell you about that. See this picture. So, in this picture they have given some dinosaur fossils: Stegosaurus, Triceratops, Brachiosaurus, Tyrannosaurus. One or two interpretations from this is the ancestor for the Triceratops is Stegosaurus. The ancestor for Tyrannosaurus is Brachiosaurus. Largest carnivorous is Tyrannosaurus, largest herbivorous is Brachiosaurus. And they also mentioned about Pteranodon. Pteranodon also extinct. Pteranodon is said to be flying reptile. And here one more organism mentioned is Archaeopteryx. Archaeopteryx fossil is saying that it is having the features of both reptiles and birds. Hence Archaeopteryx, though it is a bird, it is considered as connecting link between reptiles and birds. This is the interpretation from this particular chart. Next. So, this is about evidences from paleontology, because fossils are giving the documentary evidences. Next, evidences from embryology. See the first point in the evidences from embryology. Embryological support for evolution was proposed by Ernst Haeckel based on the observation of certain features during embryonic stage common to all vertebrates. What was the observation of Ernst Haeckel? Haeckel said embryos of all vertebrates including human develop a row of vestigial gill slits just behind the head, and it is a functional organ only in adult fish, not in other adult vertebrates. This is one point of Haeckel. One more point of Haeckel is, Haeckel explained about biogenic law. What is biogenic law? Ontogeny repeats phylogeny. So, the developmental stage of a particular organism will show some characteristics similar to its ancestors. Ontogeny means developmental stage, phylogeny means evolutionary stage. So, for that one example I can give you like tadpole. Tadpole is a developmental stage. It has a tail like fish, which is its ancestor. It has gills like fish, two-chambered heart like fish. Next, it excretes ammonia like fish. So, embryonic stage having many characteristics like adult ancestor. This is the feature explained by the Haeckel. But by careful observations, a scientist known as Ernst von Baer, he disproved the Haeckel's concept, saying, see, these are the embryos of vertebrates, early embryo of fish, salamander, tortoise, chick and human. See, early embryos are almost seems to be same. That means embryos of all vertebrates are alike in early stages. So, what Haeckel said, embryos are showing similarities with the adult ancestors, but von Baer is saying that embryos are almost similar to its embryonic ancestor, and von Baer also said embryos never pass through the adult characteristics. So, this is the embryo of a reptile, this is the adult amphibian, there are much differences. Finally, he said as development proceeds from early stages to the later stages, specialized characters appear and adult forms are different from the embryos. So, embryos passes through the adult characteristics is the point by the Haeckel, embryos never pass through the adult characteristics is the point that is proposed by the von Baer. So, these are regarding embryological evidences. Next is evidences from comparative anatomy. So, here we need to talk about two important concepts like homologous organs and analogous organs. So, what is a homology? What are homologous organs? See, homology is nothing but the organs present in the different organisms whose origin is same. Their origin is the same. Let me tell you one example here. See, forelimb of man, forelimb of cheetah, forelimb of whale, forelimb of bat. Man, cheetah, whale, bat. So, all are having a same origin because they have similar anatomy. The forelimbs consists of humerus, radius ulna, carpals, metacarpals, phalanges in all this. Hence, their anatomy is same. So, organs whose anatomy same, whose origin same, but showing different functions are said to be homologous organs. Why are they showing different functions? Because from that common ancestry, they move to different habitats for their survival. So, this is called divergent evolution, as they are moving apart from the common ancestry and adapted to different environmental conditions. So, homology explains about common ancestry, homology explains about similar anatomy but different functions. The examples of homologous organs includes the forelimbs in vertebrates, that means like mammals, cheetah, whale, bat and man. Next, vertebrate brains explains homology, vertebrate hearts explains homology, and the plant example includes thorns and tendrils in bougainvillea and cucurbita is also the example for homology. Homology explains about divergent evolution. The next concept is analogy. So, regarding analogy, the origin of those organs is different, origin different, anatomy is also different, but with those different anatomical parts, they are performing the similar functions because they adapted to the similar habitat. So, this is called convergent evolution. See, X species, Y species, Z species, they migrated to the same habitat for their survival, but their origin is different. So, the analogous organs are nothing but whose origin different, anatomy different, but performing similar functions. Examples of analogous organs includes eye of octopus and that of mammal, analogous in nature, wing of a bird and wing of an insect, analogous in nature, flipper of a dolphin and flipper of a penguin, analogous, and potato and sweet potato, they are analogous in nature. These are the examples for analogous organs. Next evidence is evidence from molecular biology. Here the evidence from molecular biology is saying that among all animals the type of cell is same, eukaryotic. Among majority of the organisms, if you see the functions of cell organelles like mitochondria function is same for all, ribosomes function is same for all, genetic code is same for all, and proteins are made up of 20 types of amino acids. Nitrogenous bases are like adenine, guanine, cytosine, thymine in all organisms. So, this kind of similarity at a molecular level is explaining the molecular homology. Similarity in the molecular level, that is molecular homology, is saying that there might be a common ancestry for all these organisms. So, this is the evidence which is coming from cell and molecular biology, showing similar components in diverse organisms, saying that there is a common ancestry for all these diverse organisms. Next, regarding natural selection, see this, man has bred selected plants and animals for agriculture, sport and horticulture, and created different breeds of dogs, different breeds of other animals like sheep, etc. by breeding program. This is a kind of artificial selection. So, what happened in artificial selection? Man produced different breeds. So, it is argued that if within hundreds of years man could create new breeds within a short period, could not nature have done the same over millions of years? Answer is yes. Since some millions of years, nature could have done this process and produced some millions of varieties of organisms. And the best example for the natural selection process is industrial melanism. For this industrial melanism, I just want to explain a point here regarding moths in some industrialized and non-industrialized areas. See, there were two types of moths, like dark winged moths and light winged moths, like black and white moths. In 1850s, in some parts of the England before the industrialization, there was no pollution. At that time, there was a rich growth of lichens on the bark of the tree, so that that bark appears light in color due to lichens, due to the absence of air pollution. You all know that lichens are the indicators of SO2 pollution. So, on this light color background, dark insects are easily visible to the birds, so that birds predated on the dark insects. So, dark insects number reduced, white insects number increased. Here I can say nature selected white winged moths. But in 1920s, after industrialization, lot of smoke and soot that is coming from the industries, it has scattered over the bark. And one more thing, the air pollution killed the lichens. So, due to the killing of lichens and scattering of all these materials over the bark, the color of the background changed to dark. So, against dark background, dark moths are not visible, but light colored moths are visible. Then birds start predating on light colored moths number decreased, dark colored moths number increased. In the first case, nature selected the light winged moths. In the second case, the nature selected dark winged moths. This is the best example. And one more point I want to tell you here, no variant is completely wiped out here. And one more thing I want to tell you, these dark moths selected after industrialization, they are not formed after industrialization. Dark moths are already there before industrialization. The dark light color are formed due to mutations during sexual reproduction. So, I can say mutations are pre-adaptive. They are already present. Whenever the situation comes, they will show their existence. So, industrial melanism is the example for natural selection, but still there is a anthropogenic action. Industrialization is done by the man. And some more examples for the selection through anthropogenic action. See this, herbicides, pesticides, some antibiotics, some drugs we are using against herbs, pests, some bacteria, and also against some eukaryotic organisms. When we are using those components, for example, when you use the antibiotic for a longer period, the antibiotic can kill the bacteria that is sensitive to the antibiotic, but it can't kill the resistant bacteria. So, when sensitive bacteria are killed, the resistant bacteria which are less in number till now, due to the death of the sensitive bacteria, now slowly the resistant bacteria can develop, that leads to the selection of a resistant bacteria. So, by using herbicides, pesticides, antibiotics, drugs, we can able to select the resistant varieties against those components. This is the selection through anthropogenic action. Industrial melanism is the example for natural selection through anthropogenic action. These are also example for the selection through anthropogenic action. And one important point I want to tell you here, evolution is not a directed process based on our sense of determinism. We can't direct the evolution. But what kind of process the evolution is? The evolution is a stochastic process. Stochastic process means the process that occur based on the chance mutations in organisms and chance events in the environment. So, whatever the mutations and whatever the events that are occurring next, they are going to decide the course of the evolution. Hence, it is said to be stochastic process. Next topic is adaptive radiation. What is adaptive radiation? To explain the adaptive radiation, they are taking the finches that are present in the Galapagos Islands, saying that Darwin's finches. See this? The definition for the adaptive radiation is from this chart. It is the evolution of different species in a given geographical area from a common ancestor in radiating manner. That is called adaptive radiation. So, what is the geographical area here? Australia. What are the different species of organisms that are evolved from a common ancestor? Marsupials. Not only marsupials, placental organisms evolution in Australia is also the example for the adaptive radiation. See this? Mouse, lemur, flying squirrel, bobcat, wolf, mole, anteater, they are all evolved in Australia from a common ancestor, showing adaptive radiation. Not only the marsupials and placentals, even Darwin's finches also showing the same pattern. Initially, there is an ancestor called seed-eating finch. From the seed-eating finch, the different finches got evolved, like cactus feeding, insect feeding, fruit feeding finches, depending on the difference of making in their beak size and also in the plumage. So, evolution of finches, adaptive radiation, marsupials, adaptive radiation, placentals also adaptive radiation. The examples for the placentals I have mentioned, the examples for the marsupials here mentioned like Tasmanian wolf, tiger cat, banded anteater, marsupial rat, kangaroo, wombat, bandicoot, koala, marsupial mole, sugar glider. These are all the examples for the marsupial radiation. Flying squirrel, flying phalange, bobcat, wolf, mole, anteater, mouse, they are all the examples for placental radiation. The important point is, when one or more adaptive radiations are there in a given geographical area, one can call it as convergent evolution. How is it? Listen. For example, see, mole is from placental evolution. Marsupial mole is from marsupial evolution. Both are in Australia only, but from two different adaptive radiations. How are they showing the convergent evolution? Because they adapted to the same mode of life. So, as organisms of two adaptive radiations, they migrated to a particular place for their survival as they adapted to same mode of life. They are showing the convergence. Hence, one can call it as convergent evolution. So, the examples are example pairs regarding the convergent evolution includes the first row is talking about placentals, second row is talking about marsupials. Mole and marsupial mole are the examples of convergent evolution, anteater and numbat, mouse and marsupial mouse, lemur and spotted cuscus, flying squirrel, flying phalanger, bobcat, Tasmanian tiger cat, wolf and Tasmanian wolf. These are all the examples for example pairs that are showing the convergent evolution. Be careful guys, only the organisms that are there in side by side boxes only showing the convergent evolution. You can't say that anteater mouse, here anteater, here mouse. These two are showing convergent evolution is wrong, because these two are showing the adaptive radiation of placentals. Marsupial mouse, spotted cuscus are showing the adaptive radiation of marsupials. But mouse, marsupial mouse convergent evolution, and it's not like mouse and spotted cuscus, are they showing convergent? No, they are not showing the convergent because they adapted to different modes of life. Only side by side boxes organisms are said to be showing the convergent evolution. Next, biological evolution. In the biological evolution, I am going to discuss about theories that are favoring the evolution. The first theory I am going to discuss is the theory of Lamarck. So, 3 points only, remember guys. Lamarck is a French botanist. The concepts that are explained by the Lamarck includes, the first one is influence of environment on organisms. The second one is use and disuse concept. Third one is inheritance of acquired characters. Whenever there is influence of environment on organisms, the needs of the organisms will change. So, according to the needs, some organs may be used more, some organs are not used. Organs used more will develop, not used will disappear. That is the concept of Lamarck. But Lamarck's theory is very old and nobody is following that theory right now. The next most widely accepted theory is the theory of Darwin. So, the Darwin's theory is nothing but origin of species by natural selection. So, what are the points that are explained by the Darwin in his Darwinian theory? Point number 1, Darwin was influenced by a book written by the Thomas Malthus. The book is On the Principles of Populations. So, what Malthus said in that book, organisms grow geometrically, resources grow arithmetically. Yes, there is geometrical growth in organisms. For example, if you see the binary fission of any organism, one organism by binary fission form 2, 2 become 4, 4 become 8, 8 become 16 at a time, that is called geometrical growth. But resources will not grow to that rate. There is a slow increase in the resources said to be arithmetic growth. This influenced Darwin a lot. And whatever the points I am going to explain regarding the Darwin, they are all Darwin's observations in his voyage. He went on a voyage in a ship called HMS Beagle, and I am going to discuss his observations now. So, the observation of Darwin, let me show you this. The first observation of Darwin is prodigality, also called overproduction. Darwin observed that many species, they tend to produce more number of progeny. That is the observation. For example, a salmon fish is having only one breeding season, but it can able to produce some millions of eggs at a time. That is overproduction only. But the second observation of Darwin is there is constancy in population. Though they want to produce more, but still the population size remains same except seasonal fluctuations. So, theoretically you may say it may increase, decrease, but practically the size of the population remain constant. Why the size of the population remain constant? Because resources are limited. That is the second observation of Darwin. The third observation is struggle for existence. As the resources are limited, definitely there will be competition within the species, between the species. It can be like intraspecific and interspecific competition. That struggle for existence leads to survival of the fittest organism. That is the third observation. The fourth observation of Darwin is universal occurrence of variations. Darwin said the population is having built-in variations by birth due to sexual reproduction. But it is not like all variations are useful. Some variations may be useful, beneficial, some variations may be harmful. Beneficial variations enable the organisms to better survive in that particular environment. Harmful variations may not help them to survive. So, beneficial variation organisms are selected, harmful variations organisms are rejected. That is the next observation of Darwin called natural selection. And once the organisms are selected with beneficial variations, they should have important property called Darwinian fitness. Darwinian fitness is nothing but reproductive fitness. When reproductive fitness is there with beneficial variations, now these variations will go to the next generations. They will accumulate generation by generation, and after several generations it may develop into a new species. That is the last observation, origin of species. So, these are the 6 important points related to the Darwin: overproduction, constancy in population, struggle for existence leading to survival of the fittest, built-in variations in population, natural selection, and origin of species. Origin of species. These are the important points related to the Darwinian theory. And what are the 2 key concepts of Darwinian theory? Natural selection and branching descent are the 2 key concepts of Darwinian theory. And one more point, important point regarding the Darwinian theory is the essence of Darwinian theory about evolution is natural selection. And one more point here is the rate of appearance of new forms is linked to the life cycle or the lifespan of organisms. Yes, bacteria can produce new varieties within hours. See this? Microbes that divide fast have the ability to multiply and become millions of individuals within hours. But the same thing to happen with a bird or with a fish, it takes many years because their lifespan is long. That is the reason why rate of appearance of new forms is linked to life cycle or the lifespan. One more important point here, they have given some microbial experiment. See this is that microbial experiment point. Assume there is a bacterial culture. Some bacteria are there. They are surviving in a medium. But when you change the medium with the antibiotic killed majority of the bacteria but left something. That leftover bacteria are said to be variant B. The killed bacteria are called variant A. What is the advantage of variant B? It has some mutations already which are pre-adaptive, that are showing resistance against antibiotic, making them to survive here. When they survive, initially they are low in number, but slowly they will expand. That leads to the selection of a resistant variety. Next, this is about Darwin's theory and Lamarck's theory. Some important points. Nature selects for fitness. Yes, survival of the fittest. One must remember that so-called fitness is based on characteristics which are inherited. Yes, there is adaptability, and the adaptability should have should have that heritability. Next, there must be a genetic basis for getting selected and evolve. Another way of saying the same thing is some organisms are better adapted. They survive, and otherwise in hostile environments. Adaptive ability is inherited. Yes, whatever the beneficial variations are there, that are going to the next generations, correct only. And what is exactly fitness meaning? Fitness is the end result of ability to adapt and get selected by nature. Next is evolution a process or the result of a process? When we talking about the success story of stories of evolution, that means when we describe the story of world, you can say evolution is a process. But when we describe story of life, we treat evolution is a consequence of process. Next theory is the mutation theory by Hugo de Vries. De Vries called mutations as saltations. He did the research about the mutations in a plant called evening primrose. What did he say? Whenever that plant is exposed to certain agents called mutagens, it will produce the plants with new characteristics in the immediate generation. Some plants with short style produced after mutation, some plants with long style, smooth leaves, like that. So saltation means a single step large variation is called saltation, nothing but mutation, explained by the de Vries in the plant called evening primrose. Evening primrose. So what are the characters of mutations? Point 1 is mutations are discontinuous. Same kind of mutation will not occur again and again. Mutations are subjected to natural selection. Mutations are random. Mutations are heritable. So sudden, random, discontinuous, heritable changes are called mutations. These mutations and their characteristics explained by the Hugo de Vries through mutation theory. So Darwinian concept is after several generations by cumulative effect a new species is coming. But according to the de Vries, a new species will come within a short time. See the difference between the de Vries and Darwin approach towards the evolution. Evolution is fast, random, directionless and based on large variations according to de Vries. But according to Darwin, evolution is slow, gradual, directional and based on small variations. So this is the point explaining about mechanism of evolution. And in the mechanism of evolution, I am going to discuss about one more important concept called Hardy-Weinberg principle. What is this principle is saying? If this principle is saying that the frequency of alleles remains same generation to generation in a population. For example, if you talk about gene pool, total number of genes present in a population is called gene pool and the frequency of those genes remains same. That means constant from generation to generation according to Hardy-Weinberg equilibrium. But it will remain same when they follow certain conditions. What are those Hardy-Weinberg conditions? Mating should be random, then allelic frequency remains same. Population should be large. There should be no natural selection, no mutation, no migration. And there should be no differential reproductive success and the members of a population should have homogeneous age. When these characteristics are there, then Hardy and Weinberg is saying that the allelic frequencies remain constant. Otherwise, any deviation in these points will result in variations and variations are the raw materials of evolution. This is the point they wanted to convey. And they have given the mathematical derivation for this equilibrium, Hardy-Weinberg equilibrium. Let me tell you about that. See, for a dominant allele we are using capital A, for a recessive allele we are using a small a. In a population, capital A's and small a's are there. The frequency of capital A can be explained with a letter called P. The frequency of small a can be explained with the letter Q. That means frequency of dominant allele is P, frequency of recessive allele is Q. But in mathematical derivation, P plus Q is equal to 1. When P plus Q is equal to 1, P plus Q whole square also is equal to 1, like P square, 2PQ and Q square is equal to 1. So what is P square, what is 2PQ, what is Q square? Because we know already what is P, what is Q. P is the dominant allelic frequency, Q is the recessive allelic frequency. For example, if you take alleles are in the form of pairs forming genotypes. See, 2 capital A's forming homozygous dominant genotype, 1 capital A, 1 small a forming heterozygous dominant phenotype, 2 small a's are forming recessive genotype. So that means there are genotypes, but how many homozygous dominant genotypes? Their frequency. The frequency of heterozygous dominant, the frequency of recessive genotypes will have letters. The frequency of homozygous dominant genotypes can be denoted with P square. The frequency of heterozygous dominant genotypes can be denoted with 2PQ, and the frequency of recessive genotypes can be denoted with Q square. So this is the mathematical derivation for allelic frequency, for genotypic frequencies. Let me explain you one numerical on this. See this, assume in a population of 1600 individuals, 256 individuals with recessive trait. That means they are Q square individuals, and they are asking how many are homozygous dominant and how many are heterozygous dominant individuals. To calculate homozygous dominant and heterozygous dominant individuals, the things we know from the question is, we know recessive individuals and we know the total population. So we can calculate the frequency of that recessive individuals. So Q square value you can calculate, Q square value is equal to frequency of homozygous recessive individuals is equal to number of homozygous recessive individuals divided by total number. So recessive individuals in the question mentioned 256, total 1600, that is 0.6. 0.6 is the value of Q square, it is not Q. How can you calculate Q value from the Q square? By making it into square root, like the Q value is 0.4. As P plus Q is equal to 1, P is equal to 1 minus Q, that is P value is 0.6. So the what was the question asked? How many are homozygous dominant individuals? How many are heterozygous dominant individuals? Homozygous dominant means P square. How can you calculate P square value? So what is the P square value? 0.6. 0.6 into 0.6, 0.36. Individuals means into total 1600, that is 576. They are homozygous dominant individuals. How can you calculate heterozygous dominant individuals? That is by 2PQ value, 2 into P value into Q value. So 2 into 0.6, 0.4, that is 0.48 into 1600, 768 individuals are heterozygous dominant. Some simple numericals are asked from this. So we better to learn these some simple numericals, because they ask these kind of questions in NEET many times. Okay. So what are the factors that are influencing the Hardy-Weinberg equilibrium? See, conditions of Hardy-Weinberg equilibrium, they maintain equilibrium. Factors affecting Hardy-Weinberg equilibrium means they disturb Hardy-Weinberg. So there are 5 important factors. Listen one by one, guys. Factors affecting Hardy-Weinberg equilibrium. They include gene migration, that is one factor. Gene migration is nothing but movement of genes from one population to the other population by migration. So whenever few members from one population migrated to the other place, definitely from the population, when they are moving out, the frequency decreases. The population into which they are added, the frequency increases. That disturbs Hardy-Weinberg equilibrium. And next one is genetic drift, very, very important. Any change that occur in the allelic frequencies by chance is called genetic drift. By chance, it is said to be genetic drift. Okay, it is of 2 types, like founder effect and bottleneck effect. So what is that any change that occur by chance? Let me tell you here one example. In founder effect, there are a small group of population called founders isolated from the members of a original population and moved out. They settled in a particular area, but before moving out, when you see the population of that original, they are all having all types of blood groups like A, B, ABO. But some people separated from that, we are calling them as founders. They established a population, and finally by generation, and if you see that population now, which is called Red Indian population, they are all having 100% O positive group. So that means creation of this O positive group population, it has happened by chance, because by chance those founders who are separated from the original population were having O positive group. So like that, if any change that occur, variation in the allelic frequency that occur by chance is said to be genetic drift. Founder effect is the example, type of genetic drift. Bottleneck effect is the type of genetic drift. Next one is, along with the gene flow and genetic drift, natural selection. Mutations, genetic recombination also affects the Hardy-Weinberg equilibrium. How mutations affect? Sudden random changes in genes is called mutations, they can affect. So during gametogenesis there is a crossing over that may result in mutations, mutations results in variations. Recombination can result in variations, they can affect the Hardy-Weinberg equilibrium. How natural selection affects the Hardy-Weinberg equilibrium? So there are three main types of natural selections. One is directional selection, second one is stabilizing selection, and third one is disruptive selection. Disruptive selection. Let me explain these natural selections in this graph. See this. The first one is explaining about stabilizing selection. In this selection, most of the individuals acquire mean character value. That means two extremes of a phenotype organisms are eliminated by the nature, only the mean character value will be preserved. When most of the individuals acquire mean character value, that is called stabilizing selection. Stabilizing selection can eliminate the two extreme phenotypes on either sides of the mean value. And it stabilizes mean character value. Example is babies' weights in England is the best example for stabilizing selection. See, the peak gets higher and narrower in stabilizing selection. When it comes to the directional selection, in the directional selection, most of the individuals acquire other than mean value. For example, short neck giraffe to long neck giraffe shifting is directional selection. Initially short but later long. Light winged moth selection to dark winged moth selection is the example for directional selection, because individuals are acquiring other than mean value. Then when it comes to the third one, disruptive selection, in the disruptive selection, mean character value individuals are removed by the nature, and the individuals are divided into two subpopulations of this type and this end. If there is no reproduction, if there is no gene flow between them, they will develop into a new species, that is called disruptive selection. So centripetal selection is stabilizing selection, centrifugal is disruptive selection. So these are the points related to this. Most of the individuals acquire mean character value, most of the individuals acquire other than mean, most of the individuals acquire peripheral values. And I already told you mutations are pre-adaptive. In microbial experiments they have explained that. So mutations are already present, but their existence will be shown whenever any situation comes. Next. So after this, let us talk about a brief account of evolution. In the brief account of evolution, we have already discussed about this. Around 2000 million years ago, first cellular forms of life appeared on earth, and the mechanism how non-cellular aggregates could evolve into cellular forms, it is not clear. Some cells released oxygen, that I told you, like oxygenic photoautotrophs. Next, see, around 350 million years ago, invertebrates formed and active. Around 350 million years ago, jawless fishes evolved. Next, around 320 million years ago, seaweeds and a few plants existed on earth. Next, one more point is, we were told that the first organisms invaded land were plants. Organisms that invaded land were plants. Next, they widespread on land when animals invaded the land. Then if you see the order of vertebrate evolution, like amphibians evolved from fishes, like see, fishes, amphibians, reptiles, aves, mammals. So amphibians evolved from fishes, reptiles evolved from amphibians, aves evolved from reptiles, mammals also evolved from reptiles. From which kind of fishes amphibians evolved? The answer is lobe-finned fishes. a group of fishes called coelacanth. They thought to be extinct, but one fish related to that they caught in 1938 in South Africa. That is called latimeria. It is said to be the connecting link between fishes and amphibians. So, from fishes amphibians evolved, but evolved amphibians are early amphibians. Early amphibians evolved into modern amphibians. Then from amphibians reptiles evolved. Initially evolved reptiles are early reptiles. From them modern reptiles evolved. From reptiles birds, from reptiles mammals evolved. This chart is explaining that only. See this chart? At the bottom they have given like early reptiles evolved during Carboniferous period on the left side. They evolved into two groups like Synapsida and Sauropsida. If you see the Synapsida yellow color line, synapsids to pelycosaurs, therapsids, and they evolved into mammals. From early reptiles mammals evolved. At the top other than mammals we have birds and modern reptiles. Birds and modern reptiles are evolved from a group called Sauropsida. So, sauropsids evolved into birds, sauropsids evolved into modern reptiles like turtles, lizards, snakes, tortoise, crocodiles. So, regarding the evolution of mammals, early reptiles, synapsids, pelycosaurs, therapsids, mammals. So, from reptiles mammals evolved, from reptiles birds evolved, from the ancient reptiles, early reptiles, modern reptiles evolved. And reptiles ruled the earth for about 200 million years. When you see the egg of reptiles compared to the amphibians, amphibian eggs are without shell, they do dry up on land, but reptiles eggs are with shell, they do not dry up on land. I said 200 million years they ruled the earth, reptiles of different sizes. See this, during that time when they were dominating on earth, giant ferns like pteridophytes were present, but they all fell to form coal deposits slowly. They formed coal deposits. Some of the land reptiles went back to water like Ichthyosaurus around probably 200 million years ago, want to lead the ancestral life like fishes. But Ichthyosaurus is an extinct reptile. It is a reptile but fish-like reptile leading the aquatic mode of life. Next, about the dinosaurs. Dinosaurs become extinct nearly 65 million years ago, maybe due to climate change, or they may evolve into birds. And from reptiles birds evolved, from reptiles mammals evolved. The initial mammals that evolved were shrews-like, very small and looking like shrews, because we found early mammal fossils, they are very small. And mammals are viviparous. Mammals are protect their unborn young inside the uterus, they are having self-consciousness. Here one example is given, like after continental drift, when South American plate joined to the North America, North American species overridden the South American species. That is one point. Due to competition, South American species become extinct. And after the continental drift, when Australian plate separated from the Asian plate, due to less competition, more marsupial evolution happened in Australia. That is one more example for continental drift. Evolution of many species Australia due to lack of competition. Extinction of many species in South of America after South American plate joined to the North America due to competition. Now let us talk about origin and evolution of man. Regarding origin of evolution of man, so there is an order in class Mammalia called Primata. In the Primata there are monkeys, apes and humans, but apes are said to be the ancestors of humans. The differences between apes and man includes important differences I want to tell you here. Regarding the jaw, apes are having a protrudable jaw called prognathous jaw, humans are having orthognathous jaw. Chin is absent in apes, chin is present in humans. Arms are longer than legs in apes, whereas it is reverse in humans. Around 15 million years ago, the primates like Dryopithecus and Ramapithecus, these are the ancient apes. They were existed. They were hairy. Next, body is covered by the hair and walked like gorillas and chimpanzees. Gorillas and chimpanzees are the modern apes, but Dryopithecus, Ramapithecus are ancient apes. Dryopithecus is more ape-like, Ramapithecus is more man-like. That is one point. Second point is, few fossils of man-like bones have been discovered in Ethiopia and Tanzania. Man-like, hominid, man-like. So, what are those bones revealed? They said They belongs to 3 to 4 million years ago. They are showing the characters like man-like primates because their fossil is showing that they walked upright and not taller than 4 feet. So, some fossils of man like bones observed 3 to 4 million years ago in East African grasslands like Ethiopia and Tanzania. See, if you see after Dryopithecus and Ramapithecus, next stage is Australopithecus. Ramapithecus is ape which is man-like, but Australopithecus is the first man stage. First man-like stage is Australopithecus. This Australopithecus is called ape man. Australopithecus existed nearly 2 million years ago in East African grasslands. Cranial capacity is around 520 cc, brain capacity. They ate fruit, hunted with stone weapons. Remember these points. First man-like existed 2 million years ago, 520 cc, ate fruit, hunted with stone weapons are the characters of Australopithecus. Next stage is Homo habilis. Homo habilis also existed 2 million years ago. The cranial capacity is around 650 to 800 cc. They did not eat meat. This stage is said to be the first human-like being, and this stage is the first tool maker. Hence, Homo habilis is said to be handyman. Followed by a next stage called Homo erectus. Existed 1.5 million years ago. Cranial capacity is around 900 cc. Probably ate meat. We can't say that certainly ate meat, probably ate meat. This is the first stage who left Africa, because the fossils of Homo erectus found in Java, which is in Asia in 1891. And one of the stage of Homo erectus is called Java man. Java man is the first stage who started using the fire for the first time. After Homo erectus, next one is Neanderthal man. Existed 1 lakh to 40,000 years ago. Cranial capacity is 1400 cc, existed in East and Central Asia. It is the first stage to use hides to cover the body. It is the first stage that buried the dead, ceremonial burial. Next is Homo sapiens. Homo sapiens arose in Africa and moved across continents and developed into distinct races. When we talk about modern man, Homo sapiens sapiens, that is modern man. Modern man arose nearly 75,000 to 10,000 years back in Ice Age. Cranial capacity of modern man on an average is 1350. 18,000 years ago, this Homo sapiens stage started cave paintings, prehistoric cave paintings. Such kind of prehistoric cave paintings we have seen recently in Bhimbetka rock shelters in Madhya Pradesh. And agriculture started around 10,000 years ago. And if you see this picture, in this picture they are saying the skull of adult human, baby human and baby chimpanzee and adult chimpanzee. If you see these three skulls, the skull of baby chimpanzee is having more similarity with the adult skull when compared to the adult chimpanzee. And finally, after all these points, the last point I would like to discuss here is agriculture came around 10,000 years back and human settlement started. The rest of what happened is part of human history of growth and decline of civilization. Civilization decline means ancient civilization declined and modern civilization started. So, with this, remember the sequence, guys: Dryopithecus, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Neanderthal man, Homo sapiens. This should be the sequence here. So, with this I have completed in detail important point discussion regarding the evolution. Thank you.